enveloped viruses. Enveloped viruses. They actually synthesize specific viral glycoproteins that are inserted into the host's cell membrane. They synthesize specific viral glycoproteins, which are the spikes. When an envelope virus exits a host cell, it actually buds out of the host cell. And during that budding process, it takes on a modified membrane as its envelope. Now, upon release during this budding process, the virus does not kill the host cell right away. This is the reason why enveloped viruses tend to cause chronic infections, like the hepatitis B virus, for example. They tend to cause chronic long-term infections. Okay. Now, as we said, that the envelope is acquired from the host cell, from the infected host cell. Now, there are two places from the infected host cell that this envelope can come from. It can either be the plasma membrane envelope or the nuclear envelope. Remember, the nucleus of a eukaryotic cell is surrounded by a lipid membrane, double layered, okay? After an enveloped virus acquires the envelope, either from a host cell plasma membrane or from the host cell nucleus, what it does to it is it modifies it with viral proteins called peplomers. Peplomers, also known as spikes. So a peplomer is actually made up of a glycoprotein. They are protrusions out of a virus. They are protrusions. They bind only to certain receptors on the host cell, and they are specific for both host specificity and viral infectivity. This is a very important thing about virus pathophysiology. Okay, these glycoproteins determine a virus host range. Host range. What does this mean, host range? It means the following, that there are viruses, because of their spikes, they are only able to infect plants. There are viruses that are only able to infect humans. There are viruses that infect humans, but they're only able to affect certain tissues of humans. For example, the polio virus infects only nerve cells because of the specific peplomers. Hepatitis virus only affects hepatocytes or liver cells because of its peplomers. The coronavirus SARS-2, okay, as what we know from it from now, at this point, from what we know, the coronavirus SARS-2, because of its glycoproteins, it can affect the heart, the lungs, and the GI tract. That's why people have symptoms vary from, stomach pain. Sometimes people lose their taste. Heart, they have heart issues now, lung, and they have obviously pneumonia-like presentation. So this is very important. This is a good picture that shows us the difference between a naked virus and a enveloped virus. Okay, now the capsid proteins of the virus, they are always encoded by the virus genome, meaning that it is the virus, not the host cell, that provides instructions for how to make these glycoproteins. That's very important. So what is the functions of the capsid or the envelope? To protect the nucleic acid and also to help introduce the viral DNA or RNA into a suitable host cell. So the capsids, now we're talking about capsids. Capsids. The capsids can come in basically three basic shapes. um, Isohedral shape, filamentous shape, or a head tail shape. Isohedral capsid basically has 20 faces. A filamentous capsid is named because it looks linear, thin, and thread like appearance. It has a rod shape or a helical shape appearance. The head tail is also known as a complex shape, and this one has a hybrid appearance. An example of a virus that has a head tail appearance is a bacteriophage. So I always give pictures and I ask to define what the capsid looks like. Is it filamentous, head tail, or